So let's look at the two branches of adaptive immunity, which consist of cell-mediated immunity and humoral immunity. So we're going to focus on the cellular branch in this video, which is, again, a branch of the adaptive defense system that involves T cells, T lymphocytes, that directly attacking and destroying or removing infected cells from the body, as well as cancerous cells or any cells that are deemed foreign through like a transplant. So if you receive an organ transplant of some kind or a tissue transplant, sometimes your T cells can attack and destroy those as well. So this is all part of this cellular or cell-mediated branch of adaptive immunity. So let's look at the different types of T cells that are going to be involved here. We've got cytotoxic T cells, helper T cells, and we also have memory T cells. These are the three types of T cells involved in cellular immunity. Now, the cytotoxic T cells are really there to help attack cells that are pathogenic. So any cells that have become infected by a pathogen or any pathogenic cells in general that are in the body will be attacked and broken down by cytotoxic T cells. Helper T cells help promote the action of cytotoxic T cells by producing chemicals that kind of recruit those cytotoxic T cells to areas of infection. They're also going to play a role in recruiting B cells, which are part of the humoral immune response, and recruiting other types of white blood cells that are more part of the innate immune response. They're going to play a big role in recruiting all of those cells to the area or the site of infection. And so I like to think of the helper T cell as kind of like the quarterback of the immune system. This is the part of the immune system that's kind of coordinating the attack as a whole and kind of keep branching all of the, or putting all of the adaptive and innate responses together so that they can all function together as a cohesive unit and help clear the pathogen more efficiently. Now, memory T cells will be formed during a first immune response to a pathogen, and they will remain in the body for a very long period of time, sometimes even for the remainder of your life, and they will be there to allow you to develop a memory of that pathogen so that if you ever encounter it again, you can develop a more heightened response more quickly and clear that pathogen much more quickly the second time that you are exposed to it when compared to the first time of exposure. So let's look at these stages of cellular immunity, and we'll notice that the first stage of cellular immunity consists of recognition. So the T cells have to be able to recognize the pathogen first before they can respond to it. Now, T cells are a little bit different from B cells because T cells have to respond to an antigen that is presented to them by another cell of the body called an antigen-presenting cell. So antigen-presenting cells could be B cells, they could be monocytes, dendritic cells. There are a lot of different types of these. But what they do is they engulf a pathogen, they digest it, and then they present part of the antigen from that pathogen on their cell surface. Now, once the T cell sees this, it can bind to it. It binds to the antigen and the self-protein that's found on the antigen-presenting cell. And once it does that, it can recognize that antigen as being foreign and start responding to it. So one of the responses is the production of these interleukins. These are chemicals that get given off to recruit other white blood cells to that area of the infection to help try to clear that pathogen more effectively. Now, that T cell, once it is able to recognize that antigen, it becomes activated. And an activated T cell will go through what's called clonal selection, where it will go through rapid rounds of cell division to make identical copies of itself or clones of itself. Some of those clones will stay as cytotoxic T cells, and those cytotoxic T cells will then go on to attack pathogenic cells by releasing certain chemicals or toxins into the surrounding area to kill those pathogenic cells, to cause them to lyse or go through apoptosis, where they break apart and die. Other T cells that are produced through clonal selection will become memory T cells. And so these will remain in your body even after you are done clearing the initial infection, and they help develop a memory of that pathogen so that if you come into contact with that pathogen again, if you're ever re-exposed to it, you have a memory of it, and now your T cells know exactly what they need to do to fight off that pathogen. They have a memory of it, and so they can go through this clonal selection process much more quickly from these memory T cells than they were able to from these activated T cells in the very beginning, because these memory T cells, again, they allow you to have a memory of the pathogen, what the pathogen does, how it acts, what it's going to be, what types of tissues it's going to be, trying to go towards. And so having that memory of how to defeat that pathogen, it encoded in the DNA of these cells, allows us to be able to remove the pathogen from the body much more quickly after subsequent exposures to it. So again, these cytotoxic T cells are really going to be there to help kill infected cells, kill disease cells, kill cancerous cells, and also to break down, unfortunately, cells that we might receive through transplants. They're also going to cause transplant rejection as well. And so these cells are going to physically break those pathogens down by delivering a lethal hit of chemicals, cytotoxic chemicals, which will help to cause apoptosis or death in that infected cell. They can also inject enzymes into these infected cells, which can also further break down the membrane of the cells and help kill them. Now, helper T cells are a little bit unique because they're going to be playing a role in both the cell-mediated response, but also the humoral response. They're going to help coordinate these two responses together and kind of link all of the immune system together, so everybody is working on the same page, and we can all, work together to clear this infection more effectively. Now, these helper T cells, like I had mentioned before, they're kind of like the quarterback of the immune system because they release chemicals called interleukins to recruit other white blood cells to the area. So they're going to recruit cytotoxic T cells, they're going to recruit B cells to the area, they're going to recruit other white blood cells involved in innate immunity, like the macrophages and the natural killer cells. They're going to recruit neutrophils to the area. Neutrophils, we know, can start phagocytosing and releasing chemicals to digest and kill these pathogens as well. So they're going to help recruit most of the immune cells to that area of infection and kind of get them activated so that they're aware that there's an infection, they're aware that there's a pathogen in the area, and they can all start trying to clear it. Again, the memory T cells, these are going to help develop a memory of that antigen so that if you're ever re-exposed to the antigen later in life, those memory T cells can mount a more rapid response to allow us to develop an army of those cytotoxic T cells more quickly and more efficiently, so that the second time that we encounter that pathogen, we may not even have any symptoms from it. We may not even know that we've encountered the pathogen. Because we cleared it so quickly from the body, we didn't actually develop any symptoms from it. Now, there are some instances where you may encounter pathogens that are very good at mutations, meaning they can change their DNA sequence and change their structure pretty rapidly. And when that happens, in cases like the flu, for instance, or influenza, you may notice that maybe you had the flu one year and you did not develop immunity to it. The reason for that is because flu viruses can mutate very quickly. They can change the way that they look to trick your immune system into thinking, oh, well, this isn't the same thing that I saw before. Now this is a completely new pathogen, and you essentially have to develop a completely new immune response to it. And so that's why, you want to get a flu shot, a flu vaccine every year, because each year that flu virus is going to mutate and change, and your body is going to have to adapt and change the way that it responds to that pathogen, because it's essentially a new virus that you're responding to. Same thing with COVID-19. It can mutate very rapidly as well, and so you typically have to get booster shots, or if you get COVID, you're not necessarily going to be immune to it. You may get it again a year later if you're introduced to it again, because COVID is going to mutate, it's going to change, and your body has to change the way that it responds to that. Now, there are some viruses like the measles. Measles is a virus that's very slowly mutating, and so if you get a measles vaccine, or if you have the measles or something like chickenpox or things like that, those types of viruses don't mutate as quickly, and so once you are exposed to those and you develop that immune response, you can use that same immune response from then on out to clear that pathogen. So you do end up with lifelong immunity typically for those types of viruses that are not able to mutate as quickly. So this is kind of showing the role of those helper T cells in immunity. Again, helper T cells have to respond to an antigen that is presented to them, just like the cytotoxic T cells do that we saw in the earlier slide. But when these helper T cells have the pathogen or the antigen exposed to them, they will start secreting chemicals into their environment. Chemicals like these macrophage activating factors. These will then attract macrophages to the area. Macrophages can engulf these pathogens and infected cells and digest them and present antigens on their cell surface. This can also increase the rate of inflammation in the area, which helps speed up the healing process too. These helper T cells will also secrete interleukins. These chemicals can recruit cells like the B cells and the T cells to the area of infection. B cells can go through clonal selection then, thanks to the stimulation from these interleukins. T cells can go through clonal selection as well. So remember, clonal selection is where we're making many, many copies of these cells so that they can help fight off these pathogens. And so this is how the helper T cells kind of tie all three of these branches of the immune system together. We've got innate immunity through the activation of those macrophages, humoral immunity through the activation of the B cells, and then cellular immunity through the activation of those T cells. Now, AIDS is a disease that is caused by a virus called human immunodeficiency virus, or HIV. And HIV is a really deadly virus because it attacks your helper T cells specifically. And remember, I said your helper T cells are like your quarterback of your immune system. They're the ones calling all of the plays and making sure that everybody is involved and everybody's doing what they need to do. All the cells of the immune system are working together to clear pathogens. So if you start killing off all of those T cells, which is what HIV does, and the T cell count decreases very drastically, it's going to really dampen your immune system to the point where it's not going to be able to clear pathogens for you effectively, and you're going to get sick very frequently, and when you do get sick, you're not going to be able to get over that illness. You're going to stay sick for a more prolonged period of time, and your illness is going to be much more severe. So AIDS, that term stands for acquired immunodeficiency syndrome, meaning you have a deficient immune system. It's not able to clear pathogens as it should be able to because you have essentially wiped out most of your helper T cells, which are there to coordinate that immune response. So people can have HIV for years before they actually have AIDS. AIDS is acquired from having HIV for a prolonged period of time, and which again is doing damage to those helper T cells. So typically your helper T cell count should be between around 600 to 1200 cells per microliter of blood. If that count falls below 200 cells per microliter, at that point, all of your branches of your immune system are going to be compromised, and you're not going to be able to fight off normal microbial infections that a healthy immune system would be able to. So things like tuberculosis or pneumonia, things like that. You're not going to be able to fight those off because you do not have a fully functioning immune system anymore. And so most people with AIDS end up dying from these other secondary infections, typically caused by bacteria or fungal infections, things like that, but things that a normal person with a healthy immune system would be able to fight off very easily. So let me know if you guys have any questions about the cell-mediated branch of immunity. We'll look at humoral immunity in a further video.